So I'm really excited to introduce Kara today. For those of you who don't know, she did her bachelor's at Penn State University and then went on to get a doctorate at the Philadelphia College of Osteopathic Medicine. She stayed in Philadelphia for her internship and residency at Thomas Jefferson University, which is affiliated with the DuPont Hospital for Children. She came to Stanford in 2007 for her fellowship in hematology oncology, and then entered the lab of Gary Nolan, who mentored her, as she developed into a really outstanding physician scientist. So Kara's been an instructor, or was appointed as an instructor in 2011. And what's really notable is that at the time we didn't have the Bass endowment, and she has sustained funding for herself and then grown that funding to establish a research program. So that's a lot of years of continuous funding, and it just demonstrates how hard work can really pay off. She is an outstanding scientist. We at the American Society of Hematology annual meeting this past December, which is probably the largest meeting for our field that occurs every year. I went to an ALL biology session and it was dominated by oral presentations from Kara Davis's group. So it was really, I felt really proud to be in the audience knowing that she's one of the faculty within the Bass Center. What I love about Kara's research program is it really beautifully demonstrates how important it is to understand basic biology concepts in order to understand how cancer develops. And she is going to talk to us today about several exciting projects that are going on in her group. And I just want to add that, you know, when I came and I was looking at the faculty, it was very clear to me that this was someone who was an incredible physician scientist, a triple threat, an outstanding clinician, an outstanding scientist, an outstanding educator. And if she hadn't been here, I'd probably be trying to recruit her right now. So I'm really thrilled to introduce her.
Thank you so much, Tanya. And thank you so much for the opportunity to be presenting today. So let me get my slides going. And as I do, I just want to also share my deep gratitude for being part of the Cohen lecture. Harvey has been a mentor for me and someone who has really helped me think about science, about taking care of children with cancer, and about being a human being. So thank you, Harvey, for all of those lessons. So today I'm going to talk to you about the research that we're doing in our lab using single cell approaches to help improve and predict patient outcomes in children with cancer. So I like to start by remembering that 100 years ago, childhood leukemia was universally fatal. And so this report from 1933 talks about the short course of disease for children that present with hepatosplenomegaly and high white blood cell counts, and the very few treatment options at that point that were available for them. But now, as we fast forward, let me make sure I can get my slides to advance. There we go. Fast forward through to the present day, these Kaplan-Meier curves really demonstrate that the treatment of childhood ALL is really a poster child for success in cancer treatment, where we have gone from very poor outcomes for children in the 60s, both at, you know, the studies on the left are from St. Jude Children's Research Hospital, and on the right, the Children's Cancer Group and Children's Oncology Cooperative Group trials up until close to the present day, and the outcomes now are quite good for the large proportion of children with ALL. However, in our patients, relapse remains a significant challenge, and this is a challenge that drives me and the work that we do in the lab, is that most children will respond to initial therapy, but relapse is the thing that will kill many patients. And so on the left-hand side, this is an old study from 2008 that was looking at whether or not there were improvements in outcomes for patients on clinical trials for relapse over the course of 15 years. And there really wasn't in these different categorizations of relapse type. And on the right here, the study from 2008 also shows that even if children achieve a second remission, meaning they're able to get back into remission after a relapse, they still overall have poor outcomes. And this even now, I was very fortunate to be part of the multi-center study of chimeric antigen receptor T cells, CAR T cells against CD19, which have been really an exciting development in immunotherapy for relapsed childhood cancer and B-cell malignancies in general. But this data also shows that about half of patients were still having events, and a lot of these are relapses. And this more recent updates of this data now with coming up to 5-year survivals look about the same, where about 50% of patients end up relapsing and not achieving a durable remission with these CAR therapies. So relapse is still a problem. And, we have developed and sort of established foundationally an approach to using single cell tools to uncover cell populations that are treatment resistant. So when a child is diagnosed or any patient is diagnosed with leukemia, they have billions of leukemia cells. We initiate treatment and can see how fast their leukemia goes back into remission where we would not be able to detect those cells. And the majority of patients are cured, but some will go on to relapse. And so foundationally, we are very interested in using single cell tools to look at the leukemia and in particular at diagnosis or before they actually receive their therapy to see if we can find cell populations that are going to translate to some sort of a clinical outcome. And in particular into cure versus not being cured. Our sort of mainstay workhorse is mass cytometry, which was just being sort of developed when I was in Gary Nolan's lab, and I was really fortunate to be there when this was happening. It's not so rare anymore, but at the time it was a very rare technique, and it brought about a lot of challenges in data analysis, which I think we can now see extend to many types of high throughput, high parameter single cell technologies. So just briefly, sort of the experimental overflow of how mass cytometry works. And I will say the other name for the instrument is the CyTOF, which I may say. And so our typical workflow is taking our cells of interest, in this case, leukemia cells. Sometimes we will stimulate them in vitro with cytokines or drugs. You can fix them and then stain them both intracellularly and on the surface of the cell with antibodies that are stained with mass tags or isotope tagged antibodies. These are injected through the instrument in a single cell spray, essentially, and then they are ionized as they go through the instrument and are measured by time of flight to the detector. The metal is measured in the time it takes to get to the detector. The data comes out looking very much like flow cytometry data. So you have, single cell events, but the benefit here is that the specific mass isotope tags of the antibodies don't have overlap in the way flow cytometry does. And so you can look at dozens of proteins on individual cells. And I quickly learned that, one of the biggest challenges was, and this is something particular to cancer, is the incredible heterogeneity when you are looking at individual cells from patients. And so the first thing we had to address was how do you organize all of this data when you are analyzing hundreds of thousands of cells from, dozens to up to hundreds of patients. And I liken it to this analogy, which I used to see more frequently at my house when my kids were little, which is, you could say that this pile of Legos is heterogeneous. And the goal of that pile of Legos is to make it into a model, for instance, of a spaceship. Oftentimes, you could also say this is a hot mess. And how am I going to get from here to our model? And the way that you do it in Lego is you do it with prior knowledge or instructions. And so you can take this pile of blocks and use your instructions and prior knowledge to build your spaceship. So the question we had then was, how do we do this with cancer data? And way back when, this is some of the first patient samples I ever ran on the instrument, when we just did some clustering approaches to try to find, and really the goal being, what are shared cell populations across these leukemia samples, which are each one of these dots is colored by the patient. What we found was it was challenging because just with unsupervised clustering, the patients mostly separated into their own subpopulations, and they didn't have a lot of overlap of cell populations that were shared between them. So this way back when was a challenge and took us quite a long time to, figure out how to approach the data. And now, we have a philosophy, I will say, that we're developing, and this is work led by Tim Keese in my lab, to approach single cell data, and not just CyTalk data, could go for any sort of single cell data, into the different levels at which you are interested in looking at that data. So either at the level of the single cells, at the level of the cluster or the subpopulation, which can be generated by many different tools. And then, of course, at the level of the sample or the patient sample or the mouse, whatever it is that you're looking at, or the condition. And that there are now a whole suite of tools in order to approach all these different levels of analysis and to move between them. But at the time, initially, when we started in ALL, we went back to the structure, or our instructions were what normal B cell development looks like. And so this was not a new concept of using that structure to sort of organize heme malignancies. However, we had some instructions already made, because one of the side projects when I was in Gary's lab that actually turned into a main project was defining the developmental trajectory of normal B cell development in human bone marrow. And so through that project on the CyTalk, we were able to really align across developmental time, which is on the x-axis of this heat map, the expression of surface markers, intracellular markers, enzymes, and align them with sort of reported fractions, mostly that have been described in mouse, and align them with regulatory signaling and rearrangement of the immunoglobulin, which of course is the main job of a developing B cell, is to make a productive immunoglobulin rearrangement. And so we took that trajectory and developed a developmental classifier for ALL. And so the way that this works is on the bottom of this figure are our healthy developmental populations, which are gated by hand the same way we would do with typical flow cytometry data. And these were gated based on what we had known from that previous work. And then each one of those populations is given a developmental barcode based on the expression of 11 proteins. And these are shown here in this heat map on the top right. Then every leukemia cell, and we did this in a cohort of 60 samples that were all taken from diagnosis. Every cell was matched to its most similar healthy, population based on expression of those 11 proteins. This enabled us to further identify cells that were predictive of relapse from the diagnostic sample. We call those relapse predictive cells. And the model that we developed was by taking those developmentally classified populations, we put them into a machine learning model, actually an elastic net model, where we developed a set of features that was predictive of relapse. And the model ended up being called the Developmentally Dependent Predictor of Relapse, or DPR is how we say it. That model is really well separated, again, from diagnostic samples, patients who would go on to relapse versus patients who would not. And actually then even further identified predictive features and the cell type that they actually happen in that are implicated in that relapse. And in this case, they really implicated active signaling downstream of the pre-B cell receptor. I'm going to talk about that a lot in future slides. And what was even more exciting for us was that the model, DPR, when we used it in comparison with our current models that we use for predicting outcomes for kids with ALL, it actually improved the performance of those models. So that, having this information from the diagnostic sample gave us more accurate prediction than our current gold standard prediction methods right now. So what's even more exciting about that is that, having that knowledge, you could actually intervene potentially earlier in therapy if you had a better way to make these predictions. So I mentioned that the model implicated a signaling network, and this is a conditional, this is like a conditional dependency model of the signaling data, where we found essentially that these three proteins, SYK, phosphoriboprotein S6 and phosphocreb were acting in a network, whereas one changed, the others changed. And the relationship between those three molecules actually strengthened between diagnosis and relapse. And if we show now back to the very, single cell level of this in two different patients, we could see that both at diagnosis and in relapse, in this specific population of pre-B cells, that there was an enrichment of these cells that have activation of all of these molecules at diagnosis that either persists to relapse, or in the second patient here example, has even more cells in that population at the time of relapse. So this really then left us with a hypothesis that these pre-B cells with active pre-BCR signaling, they characterize relapse predictive cells. And so is that signaling part of what enables them to persist and to repopulate a relapse? So I want to take a moment and go back to immunology and review, what happens across this developmental time in B cells. So B cells go from being early pro-B cells, pre-pro-B cells, and they go through these consecutive stages of differentiation where they are characterized either by cellular differentiation and they are not cycling, versus proliferative bursts where they are cycling as they are going through the different developmental steps. And there are checkpoints that dictate the progression through these stages, and one of them involves signaling through the pre-B cell receptor. The pre-B cell receptor itself is actually it's like a trying on of the immunoglobulin. So it happens after a cell rearranges its heavy chain, and it assembles with these surrogate light chain proteins where it tries on whether or not it signals properly. And if it does, you can go on and rearrange your light chain. And the marrow itself is sort of like a conveyor belt of development, so that these early cells are going from progenitor stages and they're moving through different niches in the marrow where they have access to growth factors like IL-7, where that helps them progress through this development. And the balance in these signaling networks is actually what helps to, push this progression internally in the cells. And so downstream of the pre-B cell receptor, SYK is a very important mediator, and IL-7 signaling is mediated through PI3 kinase, also the STATs, and all of these things combine with other factors, of course, to enable opening and closing of chromatin, access to things like RAG, which is actually the enzyme that will cut your DNA as you're going through rearrangement and eventually help you progress through. So pre-BCR signaling has been implicated in leukemia and in treatment resistance. And so Marcus Mushin's group actually now about 7 years ago published work showing that you could take ALL cases as a case, like a bulk, all the cells, and you could categorize them into pre-BCR positive or pre-BCR negative, and that those were characterized by different signaling networks, and that in general, the pre-BCR positive cases are pretty rare compared to the rest of leukemia, and they are enriched in some genetic subtypes. So after the initial data from our model about relapse predictive cells, we like to characterize them, and I like to think about them like this: are these relapse predictive cells that are sort of hiding in the bulk at diagnosis. They have this active pre-BCR signaling, and it's always on, constitutive on, and so the lights are always on. Versus our remission predictive cells, like the gray cells, the signaling is not active, the lights are off, but they can be turned on. You can actually stimulate the cells, they will activate the proteins downstream of the pre-BCR, and so you could turn on the lights, but they're not on all the time. And so we went back to some samples from that cohort that was published, and we sorted out the pre-B cells from 3 patients who would go on to relapse and 3 patients who would not. And so the patients that had these relapse predictive cells versus they do not, we saw differential expression between those different cell groups. And when we looked at the pathway analysis, we were interested and excited to see, and I was a little scared, that it really pointed out a lot about cellular metabolism. So oxidative phosphorylation, reactive oxygen species, glycolysis, and fatty acid metabolism all came up as enriched in our relapse predictive cells. And we also did see gene networks that were associated with the signaling networks that we had seen on the protein level. So PI3 kinase and mTOR signaling, mTORC signaling, all was also implicated in these cells that predict relapse. Dora Jadwi, who's an undergrad in the lab, went to the Broad DeepMap portal and looked at cell line data that was pre-BCR, positive or negative. The DeepMap portal uses CRISPR screens in order to characterize different cancer cell lines. And when we looked at the genes that were specific for the preBCR positive cell lines, again, we saw enrichment in pathways related to translation and energy generation, including oxphos, TCA, the TCH cycle, and a lot of mitochondrial genes. So in order to have a model to work with, Yuxuan Liu, who's a research scientist in the lab, went on to take some of our cell lines, so we had more cells to work with, and knocked down sick using CRISPR so that we had a way to sort of cut the power to these preBCR always on cells. And so this is an example of what that looked like after her knockdown in some preBCR positive lines. She also did it in some negative lines. And so we could start to look at metabolism in these cells. And so when she did RNA sequencing on the different cell lines, comparing the knockdown and the wild type, we saw that that when the wild type cells, so the cells that had the activation of the preBCR, they had gene expression that was similar to what we saw in our patient samples. So again, an enrichment in oxphos, mTOR signaling, and fatty acid metabolism. And whereas the wild type cells, or sorry, the knockdown cells were more similar to the gene expression we saw in the remission predictive cells. So this has taken us into a new direction for me, which is learning all about cellular metabolism. So going back to biochemistry, on the left, and I forgot to acknowledge, but this beautiful figure is from Lucille Stuani, who's a postdoc in the lab, who is a metabolism expert, to remind us that there are many ways and many types of substrates that you can put into your mitochondria to generate energy, as well as generate via glycolysis. And we have seen through some seahorse assays, and I'm not showing all of them here, but there are differences in the preBCR positive cell lines versus the preBCR negative cell lines based on how they use energy. And in particular, the preBCR positive cells are much more flexible in order to flip between glucose or fatty acids, which are their preferred substrates, to glutamine, compared to the preBCR negative lines. And in general, they seem to have they are more energetic in their phenotype compared to the preBCR negative lines. So this seems to make a link between this activated signaling and the energy that the cells need to maintain it. So in collaboration with Sean Bendel's lab, we've been working on developing a CyTOF panel where we can look at metabolism and signaling and development in these different cells, in single cells. Yuxuan piloted this in 9 patient-derived xenograft samples, which we categorized as preBCR positive or negative based on their expression of those proteins. And we could see that actually in the positive versus negative patient samples, and they're gated on sick, so whether or not they actually have a higher level of the protein to initiate the signaling, that there were, especially in the preBCR positive samples, that there was always a higher level of signaling downstream of the preBCR through like BTK S6, but also with mitochondrial proteins like ATP5A and IDH2, which are all involved in oxphos. And so interestingly, this is starting to mirror some things that have already been published in AML about differential metabolic flexibility and how that plays into treatment resistance. And so this is my last slide on this topic because this is a project that's ongoing, but Yuxuan has been further optimizing this approach in collaboration with Miles Lin from Ravi Majeti's lab and Rafael Aguayo, who's in Marseille now, but was previously at UCSF, to be able to sort of do similar assays to what is done on the seahorse and apply it to the CyTOF, where we can actually use metabolic proteins and the measurement of metabolic proteins in combination with ex vivo manipulations of the cells, and couple that with signaling and developmental phenotype and transcription factors so that we can look at the single cell level in patients about how these things relate to treatment resistance and patient outcomes. And we have a cohort of about 130 clinically annotated ALL samples that we hope to have this panel ready to use. So I'm going to transition and talk a little bit about a second project in the lab that is, again, about treatment resistance in ALL, and in particular, resistance to glucocorticoids. So glucocorticoids are both endogenous and exogenous that we use for many things clinically. I think sometimes I always want to remind myself that they do things, and we oftentimes don't think about exactly what they do on the cellular level, but glucocorticoids have an important role in regulating gene expression. They do this by binding to their receptor, the glucocorticoid receptor in the cytosol of the cell, which then translocates to the nucleus, and the bound glucocorticoid receptor complex can then bind and mediate changes in gene expression in 3 different ways. It can bind directly to glucocorticoid response elements in the genome. It also can bind via transcription factors, or it can do both. It can bind to response elements plus transcription factors, and in that way, it can mediate changes in gene expression. And these actually can either be sort of activating gene expression or repressing. So it can have many different functions based on the tissue and the specific context. In leukemia, glucocorticoids are really a mainstay of therapy and a backbone for ALL therapy and have been for decades. This paper demonstrates the European experience where they usually use a 1-week window. When a patient is first diagnosed, they get 1 week of steroids before they start the rest of their chemotherapy. And through the experience in those clinical trials, it's been well demonstrated that patients who have a better response to prednisone or more cell death after that week of prednisone, so prednisone good responders, have better overall outcomes than patients who have poor response to prednisone alone. I kind of think about this as the poor man's minimal residual disease measurements that we do in a much more sophisticated way now. So Yolanda Sarno in my group has been looking at glucocorticoid resistance. And so to simplify that pathway a little bit, of course, we can see that there are different pro or anti-apoptotic effects of glucocorticoids. And to kind of tie this in a little bit with the cellular metabolism story, Marcus Mushin's group, again, a few years ago, published a paper showing how derangements in normal B-cell transcription factors, including things like PAX5 and IKAROS, actually regulate the level of expression of the glucocorticoid receptor. And then that also can change from normal B cells into when you have overt leukemia and the amount of energy that is needed in the cells. So this all started actually way back when I was in Gary's lab. After we published that first paper on normal B cell development, I was really interested in understanding what were the gene expression or gene programs that changed to help mediate those changes developmentally in that differentiation. So we started out by sorting healthy bone marrow from 3 healthy donors into 3 developmental populations and performed RNA-seq. This was before single-cell RNA-seq was as easily available as it is. And when we did some pathway analysis in those experiments, what we found, interestingly, in these normal cells was that genes related to B cell receptor signaling and B cell development moved developmentally in the same way as glucocorticoid receptor signaling, which was really not expected. And, there was not really known a role for glucocorticoids in healthy B cell development. We then went to the cytoph to see if on the protein level, we could see the glucocorticoid receptor in healthy marrow, and indeed we could. Interestingly, in this heat map down here, it tracks developmentally with pro-B cells and the expression of other markers of early B cells like TDT and CD34. And then it starts to decline as cells mature and gain CD19 and CD20. We found that it was cycling cells in S-phase and G2 that expressed the highest levels of the receptor across these early B populations, and that if we treated the cells with glucocorticoids, and in particular we used dexamethasone, that we would see an inducement of cell cycle arrest, but not significant apoptosis actually in these healthy cells. So knowing that the importance of glucocorticoids in ALL therapy, we went to look at some ALL cell lines initially, NOM6 and REH cells. And we could see actually that the NOM6 had about 40% of their cells die after exposure to 2 days of dexamethasone in culture, and that there also was an impact on their cell cycle very similarly to what we saw in healthy cells. But the REH were pretty impervious. And when we went and looked at the level of expression of the GCR, it actually turns out that REH don't express a GCR very highly, and they have a mutation that limits the expression of the protein. Yolanda went and introduced the GCR back into these REH cells, and that resensitized them to glucocorticoids with similar effects on cell cycle. And when we But then we were really interested in what are the cells that actually survive the treatment with dexamethasone. And we found that the pro-B cells were the ones that were sort of preferentially killed. And I'll remind you that pro-B cells were the ones we saw with the highest level of the receptor, leaving the pre-B cells as the ones that survived. So looking again at gene expression in these surviving cells, what we found was that cell cycle pathways were downregulated in these surviving cells, but the pathways that were upregulated were interesting. And again, some familiar players. mTORC and mTOR signaling and BCR downstream events were all gene expression programs that were enriched in the cells that survived. And interestingly, we also have seen metabolism show its face here as well, with fatty acid metabolism response to hypoxia also being enriched. So it's likely that there is an interplay, not surprisingly, because of how glucocorticoids work in cellular metabolism in these early B cells. So seeing this activation of genes in the B cell receptor pathway made us think that we could potentially target this pre-B cell signaling to combine with glucocorticoid treatment to improve apoptosis. We did this in some cell lines, and indeed, when we add dasatinib to dexamethasone, we have improved cell killing in these two cell lines. And so we went on to look at patient samples, about 14 patient samples, who also had 48 hours of ex vivo treatment with these for three drugs and the combination. And again, in the patient samples, similar to the healthy cells, we see highest expression of the GCR in the pro-B cells, and it tracks again developmentally, but it was really heterogeneous. So if we look across the population of patients, again, it's highest in the pro-B cells, but there's quite a bit of spread. And interestingly, what we saw was that the patients who have clinical, we knew they were either clinical poor responders or relapse samples, had the highest level of expression of the GCR in this cell population. Looking at now how the patients responded to dexamethasone or dasatinib or the combination, we again saw overall that there was cell death with dexamethasone. It was not as much with dasatinib alone, and it was similar with the addition of dasatinib at 48 hours. If we specifically then, what we did was we looked at dexamethasone response, and we divided the patients into responders and non-responders based on having more or less than 10% cell death after the treatment. And as we had suspected, the non-responders expressed higher levels of the GCR, which is not necessarily what is known in the literature. But again, when we're focusing in on a particular cell population, in this case, these pro-B cells, they have this very high, much higher level. And when we looked at how the patients started as far as their developmental phenotype, the non-responders had more pro-B cells, excuse me, to begin with. But both the responders and non-responders after dexamethasone enriched in this more mature pre-B cell population. We saw that more formally when we used our developmental classifier. Then in both cases, we saw enrichment in the later pre-B population after treatment with dexamethasone. And when we looked at what was happening inside the cells, we saw that the surviving late pre-B cells upregulated CREB and S6 after dexamethasone treatment, and that the combined response when we added dasatinib, and I'm not showing the data here, but that actually will inhibit the preBCR signaling. But at least at 48 hours, we didn't see that had an impact on apoptosis. We had access to some patient samples that were paired from diagnosis and day 8 from patients who actually had received the window of dexamethasone treatment, actually prednisone treatment on clinical trial. And indeed, when we did the developmental classification, the day 8 sample again shows this enrichment of pre-B2 cells. And if we look at differentially expressed proteins versus diagnosis, we see activation of some of our downstream markers of the preBCR, including S6 and ERK and CREB, as well as a decline in sort of immature looking cell proteins like CD10 and TDT, and a rise in things like CD19 and CD24 as these cells appear to be maturing. Yolanda then did an in vivo experiment using the combination of dasatinib to target this preBCR signaling with steroids, because this is not something that's used clinically widely in patients. And what we saw was that The combination could reduce engraftment, and this is showing the bioluminescence data, as well as marrow and spleen engraftment. And in the purple here is the decreased engraftment with the combination in both the spleen and the marrow. And even more, we could see the blunting of the S6 and CREB activation when we added the combination that was most efficiently addressed when we had the combination. Developmental classification, again, recapitulated the change in phenotype in the cells that survive the treatment. And then also that combination did result in a survival advantage for these mice. This was in a cell line PDX model, which is very aggressive. So we actually have ongoing experiments with some of our primary samples to see if we can see this with these primary cells as well. So this left us, for this part, with a model in which we found in healthy cells that the glucocorticoid pathway coordinates with pre-B cell developmental pathways at the pre-B checkpoint, that the leukemic cells that are expanded at this pro to pre-B stage also, maintain some of the developmental expression of the glucocorticoid receptor, and that the cells that survive glucocorticoids seem to be pushed toward a more mature stage in which they may be more reliant on preBCR signaling. And if we target that signaling, we can actually improve cell death, both in vitro and in vivo. And so I'm going to turn to the last story. I think I have time to get through this, talking about cell resistance in the context now of these newer immunotherapies for relapsed ALL. I mentioned at the beginning that CAR T cells against CD19 have really been impressive for helping kids with relapsed ALL. And having had the pleasure of being part of these now for many years, it's been amazing to be able to have options for these patients. However, as with all things, we always need to do better. So many of the relapses on these trials have been CD19 negative. So the cells downregulate or take the target off the surface of the cells, and now your CAR T cells don't work anymore because the leukemia cells become invisible. There's been many now mechanisms that have been described for CD19 negative relapse after these CAR T therapies. And a couple of them that have been identified have highlighted alternative splicing of CD19 as one of the mechanisms, and that changes either the localization or the ability of the protein to be translated. So Pablo Dumisi in the lab undertook a project where we wanted to, again, see could we find cells before a patient got CAR that would predict whether or not they would have a 19 negative relapse. This is relevant not only for CD19 CAR T cells, but other small molecules targeting 19 like blinatumomab, which is very widely used in adult and pediatric oncology. And so in collaboration with some folks at Children's of Philadelphia, Dave Barrett and Steve Grupp, we had access to a lovely cohort of patient samples that had been expanded in mice and their clinical outcomes, either pre or post CD19 CAR. They were evaluated by CyTOF on our sort of standard ALL panel. And so, of course, then they went through our standard developmental classification. So one of the things that we were challenged with is that initially when Pablo was doing this, initially he just knocked out CD19 by CRISPR in some cell lines. And when we looked at developmental classification of cell lines, it didn't change very much after the loss of CD19. But when we looked at this in the context of these patient samples, what we found was that if we just directly classified the pre-CAR sample to the post-CAR sample after the loss of 19, we found the enrichment in our pro-B population. But we actually, interestingly, had this enrichment in this population of cells we call early non-B cells, which is sort of a garbage lineage negative population that we have defined. It doesn't have anything that makes us think it's a B cell. So Pablo dug into those cells. And of course, our normal developmental classifier uses CD19 to define cell developmental state. So what he did was he looked at the classifier without using 19, took it out of the definition. And what we came up with was a subpopulation of cells called proB2 discordant cells, meaning cells that actually, when we include 19 in their classification, they are called proB2. And if we take CD19 out, they're called early non-B1. And there's actually a lot of differentially expressed proteins that I'm not showing in these cells so it's not just CD19 that is changing when they're losing it. We actually found that these ProB2 discordant cells existed in every sort of category of ALL, including patients who had complete response and including de novo ALL cases, patients who had never gotten CAR and it was their first diagnostic sample, we found those cells, but we don't find them in healthy bone marrow. So they're ALL specific. So digging into that data a little bit more, these discordant cells, Pablo found that there was a difference in frequency of cells that had in their levels of PAX5, which is known to regulate CD19, and also IKAROS, which is a transcription factor I mentioned that may play a role in metabolism, that was also low in these cells. Pablo went on to do some single cell RNA sequencing in a subset of these patient samples and confirmed also at the mRNA level that in the pre-CAR sample from patients who would go on to have a 19 negative relapse, that there was this population of cells that had low levels of IKAROS mRNA. A word or two about IKAROS, what's always interesting in research when old things come around. It was one of the first things I was interested in research when I got into Gary's lab, because there were some papers that came out about its importance in ALL. And so IKAROS is a member of a family of zinc finger transcription factors, where it has a DNA binding domain, and also it usually homo or heterodimerizes with other members of the family, and that's the protein interaction domains at the end of the protein. It's very important. It plays a role in remodeling of chromatin by coupling with the SWI/SNF complex or the NURD complex. And it's important in early development, but specifically in lymphoid specification. And as I mentioned, I was going into the lab in 2008 in Gary's lab, and this paper was published about its importance in pH positive ALL. And then it's been found subsequently to be really important and of poor prognosis when it is deleted in ALL, and also potentially a germline variant of significance for ALL predisposition. So we know a lot about IKAROS and ALL. We don't know anything about it in CAR, and we don't know anything about it in regulating CD19. So Pablo asked the question whether lower IKAROS translates to lower levels of CD19. And so in 3 cell lines, he performed either shRNA knockdown or proteolysis by lenalidomide and asked if CD19 decreased. And indeed, with both methods, surface expression of CD19 declines with declining levels of IKAROS as well. So Pablo went on to take a multi-omics approach to look more deeply at why this is happening. And so again, in our 3 different cell lines with shRNA knockdown, he performed RNA-seq and attack sequencing. And what we found was that the mRNA levels of CD19 don't change after the IKAROS knockdown. However, and there's also no change at the promoter of CD19, so that it is equally accessible in the wild type or the knockdown condition. So that does not seem to be the way that IKAROS is regulating this. But what we did find was that IKAROS seemed to have, in the differentially expressed genes, changes in genes that are involved in RNA splicing. And so many different pathways here involved in RNA splicing. And this, of course, is interesting because IKAROS itself is misspliced in ALL, and that was the first description of the mutations that end up causing problems in ALL. And so when we looked then at different splice isoforms of CD19, we actually found that in the IKAROS knockdown condition, there was a higher frequency of CD19 that retains intron 10. And so intron 10, it's a misspliced, or maybe not misspliced, but alternatively spliced form of the mRNA, and that introduces stop codons. And so Pablo's doing some work to see how that translates into the protein stability. But we found this both in our data and then looking at some other public data, that there is this increased frequency of missplicing at intron 10, which has not been previously described. In addition, going more broadly into the different gene expression pathways, IKAROS, when it is knocked down, we start to see pathways being enriched in myeloid cells. And so the sort of enforcement of the lymphoid identity is lost. And this is another mechanism in which patients may relapse after a CD19 CAR, in which they change from a lymphoid cell to a myeloid cell. So it may be that IKAROS is also playing a role in those types of relapses. we wanted to know then, how does IKAROS knockdown affect the ability to respond to CAR? These are some preliminary experiments in conjunction with Robbie Maisner's lab. Maria Katerina has helped us with this. So putting in wild type versus IKAROS knockdown cells in the face of CD19 CAR T cells, we could see that the production of cytokines was impaired in our IKAROS knockdown cells, IL-2 and interferon gamma even more. And then actually the cell killing, which is done, this is done by an IncuCyte assay over 72 hours, that if we put in wild type cells that we get good killing with the, sorry, I realize my legend is missing here, but the green or the red is what to look at, that you get good cell killing with wild type IKAROS, but as you have IKAROS knockdown, you get impaired cell killing with the CD19 CAR. So together, this has left us with a model that, again, before getting a CD19 directed therapy, there may be proB2 discordant cells that are CD19 positive in the great population of leukemia cells. Hidden within those cells, if there are some that have lower levels of the transcription factor IKAROS, that these are patients that may be higher risk of loss of CD19 and a 19 negative relapse through intron 10 retention and potentially loss of B-cell identity. And Pablo's finishing up some experiments to get this ready to go in for submission. So I'm hoping today that I've been able to describe to you how we organize single cell data in cancer, and that we have gone from having this big mess of very heterogeneous data in different patients in order, and then using the sort of the lens of normal development as a way for us to then go back to that mess of cells and find the most important ones that we need in order to, do better things for patients, identify pathways or potential targetable pathways. And even more, what's been exciting for me is that I feel like I'm learning so much about how all of these different processes in cells regulate development. And so I told you today about signaling and regulation of gene expression by splicing and post-transcriptional modifications, cellular metabolism, and chromatin state. And the truth is, all of these things are interrelated in helping a cell normally develop, and that these processes are all part of that process. And that as we think about that, some of the stories that I told you today, we can sort of bin them in different parts of this graphic. And actually, there's a lot of connections here about how they may regulate to each other. And so we're very interested in and excited to continue to try to tease that apart and see how they all work together. So what's next for us and what's ongoing, I will say, because I've only given you a small slice of what's happening in the lab. So in addition to all of this work on ALL, it has necessitated us developing tools to improve the analysis of mass cytometry data, which is always a moving target. And a lot of these also are relevant to other types of single cell data. So two examples of this on the left, Ben Lowe in my group, we just published last month a new approach to batch normalization for mass cytometry data that even more excitingly enables the analysis of multiple public mass cytometry data sets together, which is something that has previously been limited, in contrast to how people use public RNA-seq data. So that tool has recently been put out there. And then I mentioned part of this before, but Tim Keyes is working on a really easily interpretable approach and code in order to approach single cell data and a philosophy of how to organize that data, which I think is very helpful, especially if for people who don't think about single cell data every day like we do. And of course, we have many more ALL samples that are waiting for analysis, and it's going to be a big summer in the Davis lab to do a lot of these samples now that we're back to full speed. We're very interested in integrating mutational data and also doing some screening to look for other targetable pathways. We are also addressing antigen loss in the context of other types of immunotherapy, including CD22 targeted immunotherapies in ALL. And so, much more work to be done there. And then I didn't have time, and someday, maybe another time, we'll talk about the work that we're working on in taking these same principles and applying them to other types of cancer, including AML and solid tumor in children. So with that, I will say thank you. Especially thank you to my lab, who has been, so wonderful. And I just love working with everybody in the lab every day. I highlighted a couple people's main projects, so Yuxuan Liu, but with a lot of help from Lucille Stuani, who is our metabolism and AML expert. Dora Jui is an undergrad who's been in the lab since she's a freshman, and has become, really a superstar. Ben Lowe, who is our computational powerhouse, Milton Merchant, who helps us with all of our mouse work. And then Yolanda, who has really worked with Yolanda now for probably a decade, and her work in glucocorticoid resistance and now MRD. Estrella is the goddess of the lab. She keeps everything running. Our lab manager couldn't live without her. And then Pablo in the CAR T-cell space, Tim doing computational and AML work. Marta and Pat are both working on solid tumors with high dimensional imaging. Crystal's looking at CNS leukemia disease, and Abhishek is a new postdoc who just started, and he's working on ALL and computational. And I wanted to highlight Adrian Delgado, who's a med student who I met at Mary's house many years ago, who still has contributed, and I still get to see him, and that's been lovely. Of course, our collaborators, our funders, and my mentors, Gary, and Harvey and Mary and Crystal and many, many more. So thanks for the opportunity to tell you about our work, and I'm happy to take questions.
Great. Okay, there are already a couple questions. The first is coming from Ben Solomon, who's asking, with increasing integration of repertoire sequencing on single cell platforms, would sequencing the pre-BCR heavy chain provide any insight into the trajectory of the clonal subpopulations?
Yeah,
Yeah.
Great question. Great question. Definitely something that we are starting to get excited about integrating. So Yolanda did a little bit of this analysis in her bulk data, so not at the single cell level in the glucocorticoid story. And we actually do see that there are some preferential loci within the immunoglobulin that seem to be differentially expressed sort of in our responding versus non-responding cells. And there has been previous work trying to understand productive versus non-productive rearrangements. I'll say our very preliminary look doesn't seem to make a difference, but we are very interested now that those approaches are available in the single cell RNA space to be able to look at those rearrangements.
Great. And the second question is from Sushma Reddy. Beautiful work, Kara. Could you speculate on whether your computational tools to predict relapse could be used in other organ systems to predict disease progression?
Yeah, thanks, Sushma. That's a great question. I think so. I mean, I think that's one of the things that, we are learning the best ways to go about this. And there are a lot of statistical complexities to be navigated. And so one thing I have learned, because I'm not a statistician and I'm not a bioinformaticist, is to make good friends with people who are, and work together hand in hand, really to try to, get at the question that you're asking, and then, designing the experiments in the right way. And I'll tell you, that's been a little bit of a learning as we go process. But certainly, it's great to have those folks working together with you. And it's challenging because, as you can imagine, you have this always like the multiple hypothesis testing problem, where you have so much data and it's likely you will find something. But then trying to understand if it's actually relevant biologically, is it actually causal, takes additional experiments to do, but these are great ways to start to generate hypotheses to chase.
Great. And then I have a question. I'm always thinking about translating to the clinic. And if you think about incorporating your BCR signaling inhibitors into a therapeutic setting, I was wondering, do you think it would be, you can imagine two scenarios where you actually don't give that up front and you actually select and try and push cells to become even more dependent on the signaling, and
Right.
then add your dasatinib, versus trying to hit them with both at once at the beginning. And I was just wondering your thoughts on that.
Great question, Tanya. So, when I thought about this, like adding dasatinib, or, we're doing some other experiments more specifically with SYK inhibitors. I think at least initially one way to translate it and what to look at would be to do it in the context of MRD. So potentially, like you say, patients who have persistent detectable cells at an early time point, to add a targeting agent to the BCR. and see if we can then compare historically to the rate of MRD clearance potentially at like the next time point, and if there is improved clearance by adding it, later. Because of course, adding these tyrosine kinase inhibitors, and we know using dasatinib in Ph positive disease does not come without a cost with toxicity. And so I wouldn't necessarily want to, trade that off for patients who may not actually need it. So I think potentially in the context of patients who demonstrate themselves to have persistent disease could be where we would intervene.
Yeah. And I guess that is related to my other question, which was, have you looked at more, and it sounds like you have more specific inhibitors, they're BTK inhibitors.
Yeah.
And do they also work, although I would imagine not as well as dasatinib, which hits multiple signaling targets?
So we haven't looked at it like as extensively in the context of glucocorticoid resistance, mostly historically, because kind of when we started the project, we weren't using all those inhibitors, but actually Yushuan's using a lot of them in the context of the metabolism story. And we are seeing that actually a particular SYK inhibitor seems to be the best at reversing the signaling as opposed to BTK, which is consistent with some older data we have where the like ibrutinib does not work as efficiently in these cells as we may expect it to, and also some other SYK inhibitors. But certainly that's part of it, is thinking about, dasatinib is a little bit like a sledgehammer in the kinase world compared to others.
Thanks, Kara. That was beautiful, beautiful talk.
Thanks, everybody.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.